Excellent. Thank you very much. There are a couple more
Thank
in the
you.
chat. Please answer. So with this,
I shall manage.
okay, I think we should move to the next talk, which is given by Dr. Daulet Kaldybekov from Al-Farabi Kazakh National University in Kazakhstan. And he's going to talk to us on improving gelatin's mucoadhesion through chemical modification with unsaturated anhydrides. Thank you. We can see the screen. If you can just make full screen. Thank you. That's fine. Please.
Good afternoon, colleagues. I'm Daulet Kaldybekov from Kazakh National University. Thank you for the introduction, Maria. This talk is a bit different, but very close to the top scope of the nanopore project. I visited Vitaly's group at the School of Trading, at the School of Pharmacy, University of Trading for 4 months last year, and had a great pleasure to work on the transmucosal drug delivery systems. In this presentation, I would like to talk about our work that was published just recently in the Journal of Biomolecules in collaboration with Vitaly's group. This work is one of the series of our publications demonstrating the synthesis modification of polymers with the specific sticky functional groups, alternative to alkyl functional groups, and they use as a polymeric excipient in transmucosal drug delivery. So this is actually the modification of a gelatin. So gelatin that we have used in our experiment was a commercially available type A gelatin from porcine skin. Actually, the gelatin is the industry one of the most important and widely used polyalphalides because of its unique physicochemical properties. The properties of gelatin depends on the method of production. Actually, it's derived from collagen. Collagen itself extracted from the bones, cartilage, skins of the slaughtered animals and the fish scales. Commercially, there are actually two types of commercially available type A and type B gelatins. Type A gelatin produced by the acid process and has an isoelectric point at pH 7 to 9. And the type B gelatin manufactured through the alkaline process exhibits an isoelectric point pH 4.5 to 5.5 respectively. As a protein-based biomaterial, gelatin has the excellent biocompatibility, biodegradability, non-toxicity, and non-immunogenicity. And it has been approved as a GRAS, generally recognized as a safe material by US and Food and Drug Administration. Gelatin readily dissolves in warm water and has the ability to form physically cross-linked thermoreversible gels, hydrogels actually, upon cooling below 23 degrees. And it exhibits a melting point close to the body temperature. These are the distinctive properties and the key factors driving its extensive application in pharmaceuticals, medicine, food, and cosmetic industries. Traditionally, gelatin has been regarded as the polymer with the poor mucoadhesive properties. Its adhesion capabilities are primarily attributed to its amphoteric nature due to weak electrostatic interactions with mucosal surfaces when compared to the strong covalent linkages. However, gelatin contains reactive sites in its structures, such as amines, carboxylic hydroxyl groups, providing good opportunities for conjugation with functional groups that can lead to changes in its physical and chemical properties. So the idea of this work was to develop gelatin-based polymeric excipients functionalized with cotinoyl, itaconyl, and metacrylyl functional groups that would have enhanced mucoadhesive properties and would stick on the nasal and vaginal cavity, and that could potentially be used in intranasal drug delivery or local treatment of various gynecological dysfunctions and infections. So gelatin in this work was modified by reacting with crotonic itaconic and metacrylic anhydrides in varying molar ratios with respect to free amines on gelatin backbone to yield cotinoylated, itaconylated, and metacrylated gelatins. The successful modification was confirmed using NMR, FDR spectroscopies, and the colorimetric TM-based assay. This is the NMR spectrum of pure gelatin to see which peak represents which amino acid is. These are the NMR spectra of modified gelatins. All four spectra displayed a characteristic signal corresponding to the protons of the aromatic acids in gelatin of about at 7.0 and 7.5. The degree of functionalization was determined by comparing the integrals of the characteristic double bond hydrogen peaks of each modified gelatin and the integration of the area corresponding to the combined peaks of the aromatic protons of phenylalanine and gelazine, where the signals serve as a reference. In the case of itaconylated gelatin, a decrease of lysine methylene signal at 3.06 ppm with an increasing amount of itaconic anhydride in the reaction confirmed the modification of lysine residues on the gelatin backbone, whereas the reaction with cotonic anhydride and metacrylic anhydride are all molar ratios with lysine signals disappeared, indicating the complete conversion of the amino groups. This can most likely to attribute to the increased reactivity of those monomers compared to itaconic anhydride. The determination of degree of functionalization values was also done using a glycine standard through TM-based assay. This is a trinitrobenzene sulfonic acid colorimetric acid assay originally developed by Habib to determine free amino groups in protein, sometimes called Habib method. The total amount of free amino groups in pure gelatin was found to be 0.434 millimole per gram. The amount of remaining free amines after gelatin modifications was then subtracted from the amount of free amino groups in native gelatin to calculate the amount of introduced unsaturated groups. The modification of the gelatin further confirmed using FDR spectroscopy. The FDR analysis of spectra clearly showed the key absorbance peaks as shown in this slide. The introduction of crotonal, itaconal, and metacrylyl moieties caused small shifts in the amide bonds of gelatin to a bit higher frequencies. An absorption band typical for vinyl groups C double bond C stretching vibration should be visible at around 1680 to 1620 per reciprocal centimeter. However, in our case, this was overlapped with the amide band, of gelatin to high frequencies. And absorption band typical for the amide. So these are the clearly shown in amide 1, amide 2, amide 3 bands. So nevertheless, the intensity of the amide 1, 2, and 3 peaks increased, which can be attributed to the incorporated amide bonds coupled with the AC double bond C stretching vibration. Overall, similar infrared spectra were acquired for the different batches of modified gelatins, suggesting the limitations of the FDR spectroscopy in confirming the effectiveness of gelatin modification when preparing different batches. As well now, gelatin is a polyamphoride. Its amphoteric nature is due to the presence of amino and carboxylic acid present in amino acids or in the macromolecular chain. At the pH lower than isoelectric point, the macromolecules of gelatin carry a positive charge. whereas at pH above isoelectric point, they are negatively charged. At the pH equal to isoelectric point, gelatin has a net zero charge, and polyamphoride chain adopts its most compact conformation, resulting in minimal viscosity in solution. It was found that the native gelatin, which we use type A, has an isoelectric point of 7.0, which is within the range given by the manufacturer. We also expect that the incorporation of unsaturated anhydrides groups into lysine moiety of gelatin would lower the isoelectric point significantly. Indeed, the introduction of unsaturated groups and the loss of lysine primary amines result in the reduction of the isoelectric point of the modified gelatin derivatives below that of the pure gelatin, which indicates a successful chemical modification. Gelatin solution in water known also exhibit zol to gel transition upon decrease in temperature. This behavior of native gelatin and its new derivatives was studied using rheological measurements. Considering the trend of the data obtained both from the melting and the gelation points can be determined by taking a crossover temperature between the storage modulus and loss modulus. The crossover temperatures is attributed to the zol to gel or gel to zol transition temperature, which indicates the transition from the elastic network formation to a solution upon heating or physically crossing the gel formation. It was found that the native gelatin exhibited a melting point of around 30 degrees Celsius and a gelation point of about 17 degrees Celsius during the heating and cooling cycles, respectively. Gelation and melting temperature of gelatin derivatives were observed to have a good correlation with the degree of functionalizations. The lower the TGL and melting points detected for the gelatin derivatives with a greater degree of functionalization indicate that the chemical modification affects the gelling properties of the gelatin. Before jumping to the experimental mechanical studies of gelatin-based materials, we evaluate the toxicity. So any kind of new chemical modifications of pharmaceutical polymers in order to improve drug delivery requires a thorough characterization of their safety and biocompatibility. We found that in the literature, there is a very limited information on the biocompatibility of materials containing cotinoyl, itaconyl groups. Therefore, we have undertaken a study of mucosal irritancy using slug mucosal irritation in vivo test. Terrestrial slugs typically release mucus to aid locomotion and prevent dehydration, and also when in contact with irritating substances. And this is the good feature. This feature could be used to evaluate biocompatibility of materials. Researchers from the University of Ghent in Belgium have previously developed and validated a toxicological assay based on the ability of slugs to release mucus in response to irritating substances such as active ingredients, cosmetics, excipients, and other formulations. As a result, when slug mucosal tissues is damaged, they release more mucus, they lose body weight, and this indication allows us to quantify and classify the irritants as non-irritating, mild, moderate, or severely irritating classifications. So as a result of the studies using the Smith slug mucosal irritation test, by measuring mucus production by the slugs exposed to the gelatin-based test materials, as well as to control substances, we have established that our modified gelatins with unsaturated groups shouldn't cause any irritation effect on mucosal surfaces at studied concentration, which is was 1.3%, as the values of the mucus production were at very low level of about 2 to 3%, both for negative and gelatin-based test materials, as there were no statistically significant difference between observed values, so the demonstrating the non-rotating nature of both gelatin and modified gelatin. So the values falls within the non-rotating level of classification. So in vitro cytotoxicity of gelatin and its modified derivatives were also studied using MTT assay with human pulmonary fibroblast cells. The negative control group consists of untreated cells and was considered as a 100% viable cells. And MTT results showed that cell viabilities are the comparable for the gelatin and its chemical modified derivatives, as there were no statistically significant difference in viability between these biopolymers against the control after 24 hours of the treatment, which indicates that the chemical modification of gelatin with unsaturated groups doesn't cause an increase in the polymer toxicity. So in order to evaluate the retention of modified and unmodified gelatin-based test materials on mucosal surfaces, we have freshly dissected porcine vaginal tissues and sheep nasal septum tissues obtained from the local abattoir, slaughterhouse. The polymers were loaded with a fluorescent dye, which is used as a model trap compound, placed on the mucosal tissue and washed with the fixed portions of either vaginal fluid stimulant or artificial nasal fluid. The result of each wash was assessed and evaluated using a flow-through method. We call it in vitro technique with the fluorescent detection. And the data were quantified with image analysis. So these are the exemplary fluorescent images of X-ray retention of formulations on porcine vaginal mucosa. Our findings indicate that among modified gelatins, metachloride gelatin demonstrated superior retention performance compared to pure gelatin. Traditionally, native gelatin itself is considered as a polymer with a poor retention capabilities. Thus, pure gelatin displayed considerably weaker adhesion to the vaginal mucosa compared to its modified counterparts. So there were no statistically significant difference was found between the crotonated and itocoanylated gelatin formulations at the end of wash of test. As a small portion of this study, we have produced microparticles based on modified and unmodified gelatins using spray drying technique loaded with the sodium fluorescent and evaluate the mucoadhesive performance. The surface morphology of these microparticles was characterized using scanning electron microscope. Microparticles presented similar morphologies. They were spherical and present a wrinkled surface structure free of crystals, pores, and cracks with a mean diameter of about 5 microns, which is the typical for common spray drying techniques. In this study, we have evaluated the potential of modified and unmodified gelatin-based spray dried microparticles as mucus formations, and they for the application in nasal drug delivery. The retention properties of these formulations were studied involving the same method as described above, with the slight modifications. The purpose of using microparticles is to facilitate the adhesion by allowing them to slowly swell and dissolve upon contact with the wet mucosal surface when applied or inhaled. All samples exhibit a reduction in determined fluorescence upon washing process, indicating the removal of the gelatin-based microparticle formulations from the mucosal surface. However, metachloride gelatin microparticles demonstrate stronger adhesion to nasal mucosa compared to other modified gelatin derivatives. Once again, pristine gelatin showed proven adhesive performance in contrast to its modified counterparts. So no statistically significant difference was observed between crotinoylated gelatin and itaconolated gelatin formulations throughout the experiment, as they displayed a similar retention profile. So overall, pristine gelatin displayed the poorest adhesive properties in comparison to its modified counterparts. The introduction of specific unsaturated cotton oil, itaconol, and metachloride functional groups into gelatin structures substantially enhanced the adhesive performance of oil formulations based on modified gelatins. The ability of chemically modified gelatins to adhere well to mucosal tissue is due to the reaction of between the unsaturated functional groups and thiol groups in cysteine-rich subdomains present in mucin through thiol and click microadhesion reaction to form covalent linkages, which occur under physiologically relevant conditions. This is the quite quick process that allows establishing improved adhesion within a reasonable period following dosage form and application on the mucosal surface. Additionally, the residual protonated primary amine groups within the modified gelatin structure could also bind with mucins through electrostatic interactions, as the pH of the artificial fluid stimulant solutions used in this study is weakly acid. So in this work, we modified gelatin with cotonic and itaconic and metachloride anhydrides. They were also characterized. The effect of modification on isoelectric point, viscosity, and thermal gelatin characteristics of gelatin were also studied. We also demonstrated that these materials are safe and has no irritating properties. We also have demonstrated that all derivatized gelatins exhibit superior properties compared to the native gelatin. So modified gelatins with unsaturated functionals could also considered as novel excipients. So we hope these modified polymers could also be used as a base material, could find application in other areas of transmucosal drug delivery, for instance, when formulated as films, gels, or micro or nanoparticles. So the complete details, you could also read this our work in recently published paper in Biomolecules. Actually, there were a lot of experiments have done, but I'm trying to fix all these studies within the time frame given in this talk. Thank you very much for your attention. I acknowledge the Horizon Nanopod project for the funding this also Science of Committee of Science of Minister of Science and Higher Education, and my collaborators, Professor Vitaly Vitalynsky, Professor Farkat Daivergenov, Dr. Sam Aspinall, Dr. Sam Roman Moiseev, Elina Moon, and Balner Jaisenbaev, who contributed in this work. Thank you very much.
Thank you very much, Daulet, for this very nice talk.